So today we will be talking about the neuroscience of gender and gender identity. And here is a timeline overview of the different papers that we're going to go over today. And we're going to start off where I'm going to start off with Schwab's famous paper on a sex difference in human brain and its relation to transsexuality that was published in 1995. Yep. And just to give a little more context about one of the researchers on the team, his name is Dick F. Schwab. He was born in December of 1944, and he was a Dutch physician and neurobiologist. And in fact, he was a professor of neurobiology at the University of Amsterdam. And that is where this study was conducted. And also he was a director of the Netherlands Institute for Brain Research until 2005. And so from the background, we can kind of have an idea that he knows what he is talking about in neurobiology. And Dick F. Schwab has actually been instrumental in paving the way and studying the neuroanatomy in relation to sexual dimorphism. And so that is why I wanted to highlight this author of the study. And next, I briefly wanted to touch upon one of the main brain regions that the study talks about. So the study specifically talks about the BSTC, which is a part of the bed nucleus striae terminalis, which is the BNST. And so this region plays an important role in controlling autonomic and behavioral responses, and it correlates with anxiety and response to threat monitoring, and it has also been found essential for sexual behavior. And this may be due to the fact that the BNST has connectivity networks with the amygdala, which has found to play a role in mood state regulation and arousal. And so the study's objective was to find a sexually dimorphic brain structure that is not influenced by sexual orientation. And the method that was used was to study the post-mortem hypothalamus of 6 male to female transsexuals for the span of 11 years. So over the span of 11 years, they collected post-mortem hypothalamus of different individuals. And an interesting finding that they found was a specific localization of somatostatin cells and vasoactive intestinal polypeptide innervation. And so basically that's to say they found a region in the BNST that had a sexually dimorphic structure, which means that there was a difference between the female and the males. And so here is a picture of where that region is. And I won't go over the details of what the other regions are, just because we want to get to the discussion portion. And so one of the things that they found was that the BSTC volume in heterosexual males were 62% greater than in heterosexual females, which shows the sexually dimorphic structure of the BSTC. And the BSTC volumes of heterosexual and homosexual males had no statistical difference. And this indicates that the BSTC volume is most likely not related to sexual orientation, which is why they did this. And also the BSTC volume in male to female transsexuals was 52% of that found in reference males. And so they found that the BSTC volume in the transsexuals were smaller than in heterosexual males or the male group counterparts. And also, a side note, they also found that the male to female transsexuals had smaller volumes of the BSTC than the female group, but the difference didn't have any statistical difference. And so here we can see 4 different pictures. And the first one is the BSTC section innervated by the vasoactive intestinal polypeptide and of the heterosexual man. So this one is A is of the heterosexual man, B is of heterosexual woman, and then C is of homosexual man, and D is of the male to female transsexual. So you can clearly see a difference in the volume, and you can see that by the intensity of staining shown here. And so I just wanted to point our attention to another graph, which this is a graphical representation of the staining that was shown previously. And the M here stands for males, and then the HN stands for homosexual males, the F stands for females, and TM stands for transsexuals, from male to female. And so also another thing I just wanted to mention is that people with abnormal sex hormone levels were numbered from S1 through S4 over here. And another important fact about the study was that all the transsexuals were treated with estrogen. So they hypothesized that high levels of circulating estrogen may explain why the BSTC volume is similar to that of the female groups in the transsexuals, as shown over here. However, some refutations against this was supported by the evidence that one of the transsexuals, T2, actually stopped taking estrogen 15 months before death. And remember, this was the hypothalamus of a postmortem brain. So T2 stopped taking estrogen 15 months before death because her prolactin levels were too high, and T3 stopped taking estrogen 3 months before death, which may not really support the idea that circulating estrogen may be the reason to the lowered BSTC volume. And so also, another important thing to note is that individuals T1 through T3 all received an orchidectomy, which is a procedure to have one or more of the testicles removed. And so this may have led to a decreased level of androgens in those individuals. However, another important fact to note is that T4 actually did not receive an orchidectomy, and yet the levels of the BSTC were still lower than of the male counterpart group. So those are just some things to note. Overall, though, the conclusion that the researchers made were that the interaction of brain structures with sex hormones may result in an altered volume of the BSTC, which can then help determine the gender identity of an individual. And so, as mentioned before, there are a lot of refuting arguments, some evidence that may not exactly support this conclusion. However, something that I just wanted to mention is that this paper was overall very profound in the scientific community because of its publication in the renowned Nature paper, which started the conversation that gender dysphoria could actually have an underlying neural basis. Because up until this point, a lot of people have thought of gender to be more of a socialized construct. However, this indicates that there actually may be partially a biological difference on a cellular level that does affect how a person perceives themselves in terms of their gender identity. And so this study was profound in that it opened the window to a discussion and an idea that there may be a neurologic component to gender dysphoria, and that in fact gender identity may not just be a conscious decision that it's made at one point in an individual's life. So when an individual chooses to go from a male to female or female to male, many people thought of this as just a conscious decision that an individual makes one day. However, this indicates that that may not be the case and that there is an actual biologic underlying component as to why someone may feel like they are not they're not identified as a gender that they feel that they are. And so with that being said, the same year, Professor S. Mark Breedlove was actually invited to write a commentary paper on the previous paper mentioned. And so just to give a quick background about Mark Breedlove, he received a BA in psychology from Yale, and he received his PhD in UCLA in 1982. And he taught a psychology and biology of human sexuality course at MSU. And his research was focused on the steroid hormones, on developing an adult nervous system, and the effect of steroid hormones on sexual differentiation and plasticity activation. And so he was actually invited to write a commentary paper because in the scientific community, he is also one of the well-known researchers in this field. And so he wrote a paper called Another Important Organ, if you guys found it in the folder. And in it, he raises a lot of interesting questions. And I just wanted to point out two brief ones, two brief questions that I thought were very intriguing. One was that Breedlove expressed doubts about whether there is, in fact, a correlational relationship at all between the volume of BSTC and the transsexuality of individuals, or whether it was just a coincidence. Because something that he mentioned was that the study that was done by Swag's group was a very limited study of only 11 postmortem individuals, or 6 transsexual postmortems. So yeah, he raised the question of whether there even is a correlational relationship. And the second question that Breedlove raises is whether the findings in the paper was a causal relationship, because at the end of Swag's paper, it seemed like they were trying to come up with a causal relationship, that the volume of the BSTC may result in one person identifying themselves as one gender over the other. However, Breedlove raises the question of whether it may be, if there is in fact a correlational relationship between the volume of the BSTC and gender identity, which one is the cause and which one is the effect. And he calls it the chicken or egg dilemma, as mentioned in the paper, because to point out that we don't know if it's the brain structure that came first, the difference in the brain structure that came first, or the difference in the behavior that resulted in a difference in the brain structure. And so I thought that was really interesting. And just to close off my section of this presentation, I think these findings are very interesting, but I just wanted to point out that yeah, as interesting as these papers are, there's still a lot of research to be done. And I think what's really important about these papers is that it gives us a platform, a foundation to actually have a discussion on whether we believe that there is a biological basis for gender dysphoria, and if there is, how we can go about to proceed with follow-up studies to support or refute the previous studies that have been done, so we can get more information on this topic. So yeah, thank you for tuning into my section of the presentation. I hope you enjoy the rest.